Awesome. Cool. Great. It's great to be here. Thanks, Kevin. My name is Brian He, and today I'm going to be talking about how to leverage, a class of algorithms called neural language models. And as the name kind of suggests, these were originally developed in the context of natural language processing. But instead, I'll be talking about how to apply them to a completely different set of applications. In this case, predicting and modeling evolution. And it turns out that these language models can be quite powerful in doing so. So this is joint work with some great co-authors and was done at two different institutions, MIT, where I did my graduate work, and Stanford, where I'm currently a science fellow. All right, so let's get started. To begin, I just want to motivate this talk with this really high-level question, which is this pretty fundamental question in biology, which is, how predictable is evolution? So on one end of the spectrum is this thought experiment by Gould, where you rerun the evolutionary process, starting with the universal common ancestor again, but you rerun that process for all life on Earth, and say Gould hypothesized that evolution is completely determined by chance. If you rerun this process, you'd get these fantastic new organisms with different kinds of life that don't resemble anything that we see on Earth today. But on the other end of the spectrum is this hypothesis by Morris, where actually evolution is so constrained by intrinsic rules that if you reran that evolutionary experiment, you would get back humans or a species very similar to humans. And so two ends of the spectrum, where on this end of the spectrum, there are intrinsic rules that are possible to learn, and in doing so to be able to predict evolution. And so this is kind of the high level framing of this talk. And so for the majority of the talk, I'll be describing some recent work on trying to predict and model the evolution of viral surface proteins. And then towards the tail end of the talk, I'll kind of devote a little bit of time to some unpublished work on how to extend some of these ideas beyond viruses to generic proteins and proteins evolving over much longer evolutionary timescales. But to begin, I'll be talking about predicting viral evolution, and I think the practical application of this is pretty straightforward, given that we're in the midst of a global pandemic, and in particular a particularly bad or challenging problem in developing antivirals and vaccines is this problem of viral escape. So say you have initial immune recognition of this viral surface protein, and this antibody will bind and neutralize the virus, and this host will have immunity to the virus. But what viruses can do is that over many generations, they can mutate their surface proteins so that, say, this neutralizing antibody no longer binds, and then this viral particle is then free to infect the host again. And this is the problem of immune escape. And so viral escape is a pretty big problem. It's the reason why we don't have a universal vaccine for influenza, why you need a yearly flu shot. It's the reason why we don't have any effective vaccine at all for HIV, despite decades of intense research effort. And there are questions about the durability of vaccine protection for SARS-CoV-2. At the moment, it seems that the vaccine-induced immunity is the virus. It's holding up well against the current variants of the virus. But as SARS-CoV-2 continues to evolve, there's definitely questions about whether or not it will start to escape the immunity that's generated by even the best vaccines we have today. So how do we model viral escape? Using kind of algorithmic techniques. An initial kind of key insight that we had is that small changes can have big semantic effects. And so what do I mean by this? So say you have this sentence, "The boy pats the dog," and I can actually make a single character change to the sentence that completely alters its meaning. So this is a very different sentence, but in terms of the characters, I just mutated a single character, a P to an E in this case. And actually, a similar phenomenon happens in the case of viral escape. So actually, if you mutate, say, this histidine to a serine on this influenza hemagglutinin surface protein, then this broadly neutralizing antibody no longer binds, and this surface protein can, or this viral particle, if it had this mutation, could, say, escape from this neutralizing antibody. And so an initial kind of question that we had is there something kind of related between the way that a virus can escape the immune system and the way we can model semantic change using machine learning? So to make this a little more concrete, this is how we're kind of thinking about the connection between kind of the language of viral escape. So say you have some initial protein and it has some initial antibody that's recognizing it. So if I'm this virus and I want to escape from this antibody pressure, what do I want to do? So number one, I don't want to mutate myself so that I die or that the protein misfolds or I lose infectivity. So I want any mutation that I make to my surface proteins to preserve some notion of acceptability or validity or grammaticality. But what I also don't want to do is I don't want to mutate my surface protein so that it just looks pretty much the same to the immune system and I still get recognized by this antibody. So kind of a functionally neutral or functionally maybe synonymous mutation. I also want to avoid this regime. Instead, what I want to do is I want to preserve my ability to infect cells, but I want to substantially alter my surface protein so that I can kind of escape from this antibody pressure. So this is how we're thinking about viral escape, kind of preserving a degree of acceptability or viability or grammaticality, but also being substantially altered in some way or substantial maybe semantic change. And to kind of formalize this a little more, this is the kind of key kind of word game that we introduce to kind of model viral escape. And it's what we call a constrained semantic change search, or CSCS. And so it's a pretty simple kind of word game. You're given a sequence of tokens from some language, and your goal is to find the single token change that, number one, induces the largest semantic change to that sentence, but these changes have to be constrained. They have to fit within some set of rules or grammar of this language. And this game is given in generality, so this could be a sequence of words from an English sentence or a sequence of amino acid residues from a viral protein. How do we implement this CSCS model? Well, we can use these algorithms called language models, and at a high level, what they do is they'll assign, in this masked language modeling case, they'll assign a probability to some token appearing within some sequence context. So how probable is some amino acid residue appearing at this position given the rest of the amino acids in that sequence? And when you train this language model on, say, thousands of infectious viral sequences, the output probability will begin to model something about the validity of mutation or the grammaticality of a given mutation. So that's the first part of CSCS, right? We want to learn the grammar of the language. But the second part is we also want mutations that induce high semantic change. So to model semantic change, we leverage this idea from, again, borrowed from kind of natural processing and Elmo and these models, where you can look at the distance in the semantic embedding space. So this is the output of one of the hidden layers of the language model. And it's a continuous embedding, and kind of sequences that are closer in this semantic embedding space, say via the Euclidean distance, are also more semantically similar. And as that distance increases, your semantic dissimilarity between those two sequences also increases. Great, so now we have a way to model grammaticality and semantic change. And our CSCS objective is actually quite simple. So we rank over all possible single token mutations to the sequence. We rank them by the grammaticality, and then we rank them by semantic change. And then we just acquire based on high grammaticality and high semantic change. Of course, you can imagine other potential objectives, but this is the objective that that we use. It's a pretty simple one. So this is a bit abstract, but just to root this in some intuition, we actually trained a language model on news headlines. It's a word level language model. And so here are some real, CSCS changes. So for example, we have this headline, Australian dead in Bali. Now, if we ask the language model, what is the semantically closest sequence that's just one word away, we get this mutation, Aussie dead in Bali. So this is pretty, synonymous with the original headline. But when we ask for the CSCS proposed change, we get Australian ballet in Bali. And so you can imagine that this is a valid headline. You could see this appearing, Australian ballet in Bali. But all of a sudden, it's a lot more positive. I guess depending on your opinion of ballet, from the original headline. So some other examples: blast off of Apollo 8 gets turned into blast victims of Apollo 8, all of a sudden a lot more negative. This is another kind of strange headline to begin with. Excuse me, you left a gorilla suit on the bus gets turned into excuse me, you left a gorilla killer on the bus. So a lot more negative and more strange. You can kind of do this all day, but I'll give one more example. Wine growers revel in good season gets turned into wine growers revel in flu season. And so when we kind of stepped back and thought about this, what we're actually doing is quite similar to these games, Mad Libs or Apples to Apples or Cards Against Humanity, if you're familiar with these games. And the reason why we find these games so humorous is that actually what they're doing is they're enacting this constrained semantic change search. So the rules of the game constrain the word choices that you provide the game to be grammatically correct. And the reason why we find these games amusing or funny is this semantically the semantically jarring at times word choices that we make when we play these games. So we kind of stumbled on a kind of formalization of Mad Libs or Cards Against Humanity. But this is, somewhat a toy example just for intuition. What we were actually interested in is, applying this to see if we can actually model viral escape. And we can actually just train a language model instead of on sequences of words. We can just train, using the same techniques, a language model on sequences of amino acid residues. And so when we train a language model, say on about 50,000 flu hemagglutinin sequences in the influenza research database, and we visualize that language model embedding, it's a very high dimensional vector in 2 dimensions that try to capture as much of the variation in that high dimensional embedding space as possible. And we plot them in 2 dimensions. You can see that each point here corresponds to a sequence. You can see that the sequences start to cluster according to their known influenza subtype and their host species. So what this is showing is that these language models are learning an internal representation that captures something about the antigenic similarity between these sequences. It's not learning something completely random. It's learning these kind of antigenic patterns consistent with our hypothesis. Again, if we train a language model on the HIV envelope surface protein or coronavirus spike, homologous sequences. In the envelope case, again, these sequences will cluster by subtype. And in the coronavirus spike case, these well-known human coronaviruses actually cluster with the hypothesized and zoonotic origin of these human coronaviruses. So SARS-CoV-1 clusters with bat and civet, MERS-CoV clusters with camel, and then SARS-CoV-2 clusters with these bat and pangolin sequences consistent with their kind of known or hypothesized zoonotic origin. Great. So we know that these language models are learning something about kind of the antigenic distribution of viral evolution, but can this actually predict escape? So to leverage, to kind of validate a model for predicting escape, we can leverage these deep mutational scan data sets. In this case, this is from a paper by Jesse Bloom's group at Fred Hutch, where you can make all single amino acid residue changes to hemagglutinin, and then grow up this virus in the presence of neutralizing antibody or human sera. and then infect cells, sequence these the viruses that make it inside these cells, and then compare to a control setting to find differentially selected mutations that will escape from this immune pressure. And in this way, it's kind of a very high throughput way to identify all single amino acid escape mutations to a viral surface protein. And rather strikingly, when we plot all possible single amino acid residue changes to a surface protein, which is each dot here in this two-dimensional plot corresponds to a single mutation. And when we plot them according to grammaticality on the x-axis and semantic change on the y-axis, and then we also overlay the dots with the known escape mutations from that deep mutational scan, you can see that the escape mutations kind of exclusively cluster in this high grammaticality and high semantic change quadrant of this kind of two-dimensional grid. And if we quantify this, we can also demonstrate that this escape enrichment is statistically significant. And so this is a great validation that how we're thinking about grammaticality and semantic change is actually aligning with what we see in this kind of in vitro model. And I just like to emphasize that this language model that was used to rank grammaticality and semantic change was trained on just on hemagglutinin sequences. We did not provide any explicit information on escape mutations. It just had to learn these patterns from the data. If you visualize this escape enrichment on the three-dimensional structure of these surface proteins, you see that there's a lot more escape potential in the head of influenza hemagglutinin, for example, than the stalk region. And this is consistent with the known kind of escape strategy of influenza. And for SARS-CoV-2 spike, if you train a spike language model, we again see significant escape potential in the receptor binding domain and the N-terminal domain. And when we made these predictions in, I guess, July 2020, it was kind of heartening to see that in vitro models, these two domains were the ones that were kind of showing up for escape mutations, in particular the NTD deletions and kind of loop insertions in the NTD are kind of highly suggestive of escape from antibody. So you're probably wondering if this model would be predictive of emerging SARS-CoV-2 variants. And indeed, we did do this analysis in a follow-up study. So in this follow-up study led by Cyrus Mayer at Vir, we can actually show that spike language model features are predictive of mutations that arise in circulating coronavirus variants up to even 4 months in advance with a high AUC of around 0.8. And so this is kind of an additional validation that the evolutionary information that's picked up by these language models, and in particular language model grammaticality, can be predictive of mutations that preserve viral fitness even across different waves of the coronavirus pandemic. In particular, especially this one mutation, S494P, that gets highlighted by our language model and other evolutionary features, does have experimental evidence of reducing sensitivity to these neutralizing antibodies that have emergency use approval for SARS-CoV-2. And so this is experimental confirmation that some of these escape mutations are in fact reducing sensitivity to antibody therapeutics. Right. So that was how we can kind of apply language models to viral evolution. In the last, say, maybe like 3 to 5 minutes of this talk, I'll just briefly go over how these ideas can expand to incorporate more than viral evolution, but generic proteins and proteins that are evolving over much longer timescales than SARS-CoV-2 spike or even hemagglutinin. And so we know that these language models can predict local evolution or single amino acid changes. But a big open question is, what about mutations that are more complicated, like more mutations due to recombination or more than single residue mutations? So a key insight that we had is that you can actually gain some insight into the global landscape of evolution by composing a bunch of these local predictions together. And the kind of agglomeration of these local predictions gives you some an approximate insight into the global kind of landscape of evolution. Conceptually, this is kind of similar to RNA velocity, if you're familiar with that. If not, it's not a big deal, but RNA velocity, you just try to predict locally the transcriptome that is nearest to you in the future state via splicing kinetics. And then these local transcriptomic predictions can be assembled across an entire trajectory to gain insight into the full directionality of, say, cellular development. From an evolutionary perspective, this is also similar to ideas of the fitness landscape from Sewell Wright and others, where a protein is just making locally optimal improvements as it evolves, and in doing so, it's exploring it's doing hill climbing in this fitness landscape. So in particular, we're interested in these universal protein language models, where you train a very large neural network language model on all sequences, say in UNIREF 50, and in doing so, we're hoping that this is a language model by Rives et al. From Facebook AI research, and when you train a language model on 3 million sequences, hopefully it's learning something more general about the evolutionary rules involved in what makes, say, a valid protein. So when we apply this language model to kind of stitch together the flow of evolution through this landscape of, in this case, influenza nucleoprotein, what we find is, these aren't kind of stream plots of wind moving through a 2D landscape, but in actuality, this is kind of like the flow of evolution across this mutational landscape of nucleoprotein. And when we visualize this, we can see that the actual flow of evolution, as predicted by the language model, corresponds to the known temporal evolution of the nucleoprotein. And we can predict the roots of the major pandemics of influenza, and in particular, this one well-established phylogenetic path of nucleoprotein does indeed move according to predicted directionality of evolution by these language models. So you can kind of visualize the flow of evolution occurring across this protein landscape. And because we're using these universal protein language models, we can apply this beyond viral evolution. So this is what the kind of flow of evolution looks like for cytochrome C. And what we've found pretty interesting is the predicted pseudo time, if you order based on this flow, recapitulates the ordering of these taxonomic categories in the fossil record. So it's kind of nice validation that the ordering information from this velocity of evolution is being recovered. And we can also show that this extends to even kind of the longest evolutionary trajectories, spanning all 3 domains of life, bacteria, archaea, and eukaryota. And we can also similarly kind of predict the directionality of evolution and model this flow using these universal protein language models. So to go back to that initial question, there's probably a lot in evolution that's just due to chance. I'm not claiming that it's completely constrained. There's a lot of environmental factors involved. But as Russell Schwartz and others have so clearly demonstrated during this session, there is also a lot of inherent rules that are constraining evolution, and that's what makes these language models probably particularly powerful in being able to extract these rules just directly from large corpuses of sequences. And so it's an exciting new area, the ability to maybe provide some insight in predicting evolution just from sequence data alone. There's a lot that I omitted just due to the sake of time. So these are the references describing this work in more detail if you're interested. And I'd like to thank you all for your attention. This work is done with an amazing group of co-authors. Ellen was a collaborator on the viral project, in collaboration or under the supervision of Brian Bryson and Bonnie Berger, all at MIT. And then the velocity work is done with Kevin Yang at Microsoft Research and Peter Kim at Stanford. So thank you.
Terrific. Thank you very much, Brian, for an excellent talk. We have a couple of questions on Slido. For the sake of time, I'm going to try to glom them together into one question. And it relates to porting over this idea of grammaticality to viral sequences. And kind of the related question is you're substituting letters, and in your diagrams, you appear to be substituting amino acids. But in reality, the changes that happen in virus sequences happen at the nucleotide level, the base level. So you have, I don't know how would I describe it, like a Hamming distance sort of thing to take into consideration about how many changes you would need to swap certain amino acids. So could you give a little bit of more detail on how those are implemented in your the language model?
Yeah, what's great about these language models is that they're general. So you can use any kind of alphabet in your language. So in our particular language model, we give it an amino acid alphabet, and we just break the language, the sequence up according to amino acids. But you could just as easily train a nucleotide level language model, where you just use 4 characters in the alphabet. You could also just easily train a larger scale language model, or maybe 5 MERS or something are your alphabet, or the level of genes, a gene level language model or something. So it can pretty kind of easily incorporate any arbitrary alphabet, which is a nice feature.
Okay, fantastic. I think we'll stop it there for the sake of time. We landed right at the end of this session and the start of the poster session for Translational Biology and Machine Learning Part 2. Aidong and I thank you very much for attending this session.